Okay, so we're going to start with protein-ligand interactions. Um, so for protein-ligand interactions, you want to make sure that you know that a ligand is anything a protein binds to. We're not going to go into the real specifics of what a ligand could be. Just know that it is anything the protein is going to bind to and make a bond with. So with that, interactions don't always involve a binding pocket. It can be a flat interactive surface. So also in a protein ligand interaction, it is going to be specific on what protein and ligand combination it's going to be. So the protein needs to be oriented correctly, as well as the amino acids exposed from the binding pocket. So if you look at this image right here, we have cyclic AMP, and you see that it is binding to the amino acid side chains, like in the binding pocket. So if you look at cyclic AMP, one of the things you can think about is some of the amino acids that are exposed on cyclic AMP aren't the same on each side. Cyclic AMP isn't a symmetrical protein. And because of that, it's not going to bind with the same things on each side. So that's why if you look at where serine is, it's closer to the nitrogens and stuff. It's going to make those bonds with the hydrogen and the oxygen. Say if you replaced an amino acid with serine, like it might not bind in the same way because you're switching amino acids. So basically, this is kind of like your check with R groups. You want to make sure that you know that R groups are going to be very specific with the types of bonds that you're going to make, and that goes with orientation, because I'm sorry. Is it better now? Hello?
I think it's only happening when you're talking like long sentences.
Oh, I'm so sorry. Is it good now?
Yeah,
Okay. How much did you hear of that so I can
not much.
Oh, okay.
I only heard orientation and that's it.
Okay. So, main idea is that the orientation of the protein matters. Because if you look at something like cyclic AMP, the amino acids projecting from that aren't the same on each side, which means that amino acids that bond to it are going to be different depending on the location of where the projections are on cyclic AMP. So if you swapped out an amino acid, it might not work in the same place. So you want to remember how specific your R group interactions are going to be. If you change, okay, so there's a question. If you change the amino acid, would the cyclic AMP changes orientation? I feel like that depends. If it's a similar amino acid in the same category, it might act the same. It would probably, Michelle, do you think it would affect just like the bonding in they probably just wouldn't bond, right?
Sorry, I can't see the question from the chat. Can you just repeat what it said?
It says, if you change the amino acid, would the cyclic AMP orient? I think it means like reorient. Would it change its orientation?
If you changed any of those amino acids there?
Yeah. Would the cyclic AMP reorient itself?
I'm not sure about reorient, but changing the amino acids depending on size and chemistry can affect the strength of the bond and type of bond it would make.
Yeah. And then it would also like affect the structure, and that goes into like the functioning of it. So Laura is big on structure function relationships. So you want to consider if you change an amino acid, then that's going to be a change in structure. And then how does that affect the bonding? And what could that possibly do to the amino acids? That sort of thing. I think we can go on to the next one.
Okay, so we also talked about, we also talked about affinity, and affinity describes the strength of interactions between proteins and its ligand. Can you hear me?
Yeah, but the slide didn't change.
Oh, I'm so sorry. Let me see. Can you see now?
Yeah.
Okay. So you heard everything I said, right? You just couldn't see.
Yep.
Okay. So the affinity is dependent on the strength of the bond and how many bonds there are between a protein and a ligand. And it's always good to consider the angle of the bond and the type of bond that exists. Are there any questions? No? Okay.
So when you talk about affinity, that is when you want the protein, affinity wants the protein and the ligand to bond together. That is what affinity is. Now, in contrast to that, we have your dissociation constant, that is Kd. And that is a way that we can measure the protein ligand interactions based on how often the protein and the ligand can separate. It's based on that titration experiment that Laura kind of went through during the lecture, but like that's not kind of the main idea. Just focus on kind of knowing what it is. So dissociation wants the protein and ligand to separate. It's going to be a measure of how that happens. So concentration of the ligand when half the binding sites in the protein are filled, that you want to assume is when your protein P equals C. So one thing to note is that there is an inverse relationship. They're going to be opposites, Kd and affinity. So when you have a smaller Kd, that means you're going to have a tighter protein and ligand binding interaction. That is your affinity. Because if you have a smaller dissociation constant, that means nothing is making the protein and the ligand separate from each other. So you're going to have a higher affinity that's going to keep your protein and your ligand bonded together. So yeah, that kind of know this relationship. And then there will be things like, how does this affect your concentrations a little bit? Kind of, you don't have to do any calculations, but if you look at like the proportions, like if your concentration of ligand were to increase, then you would know that it is proportionally, like directly proportional to your KD.
So
But
we're just responsible to know how this affects the cell overall?
you want to focus on kind of a big thing is the inverse relationship that you want
Okay.
to know. You want to know that a lower KD equals a higher affinity.
Okay.
And this will tie into everything that we focus on with reactions and stuff, because we have another slide on it, but it's like you don't want to do anything that is going to hold up a reaction. It's all kind of going to be regulated with the amount that things are bound together and the amount of times that they dissociate. So Michelle, I think it will be clearer if we go on to the next slide.
Yeah, I think so too. But also, guys, you're not going to be tested on mathematics. Like, I don't think she's going to give you numbers like for concentration and you calculate KD. That's not the idea with this at all. It's like May said, it's the relationship between affinity and KD.
So definitely know the definitions of each of those and know that they're opposite and they have the inverse relationship, because like when you have a reaction, like if it's going to dissociate, then obviously then if things are separating, they're not going to have a high affinity because they're not coming together, and then vice versa. So then I think
okay, I'm going to click the next one. So here's just an analogy to think about when we're talking about KD. So just think of enzymes, which we'll get to in the next few slides. But ideally, if you want an enzyme to bind to a substrate, that's going to be a very strong strong interaction. And during this strong reaction, you will have high affinity. And then because the affinity is high, then the KD is low. And this is because during the beginning of a reaction, you want the enzyme to be attached to the substrate so that the reaction can occur. And you really want it to bind tightly to the substrate so that the reaction can be performed. And then at the end of the reaction, you get product that forms. And the enzyme at this point should be able to remove itself from the product molecule and separate itself. And so at this point, you'd want to have a low affinity and then a higher dissociation constant. So you want that number to be a lot higher because you want the enzyme to be far away and you want it to completely dissociate. And this is important for enzymes because enzymes have to be used again. And if it's stuck to the product, then we can't use it again for other reactions. And yeah, so this is just something I stuck in here to help with KD and figuring out the relationship between KD and affinity. Does anybody have any questions?
I have a quick question. So she talked about like the Goldilocks hypothesis, like earlier in like the week. Does this like apply here too?
It can. So if you think about a reaction, you're starting with reactants and you're ending with products. And you don't want to make too much of that product, but you don't want to have too little of that product. So it will kind of be like a regulation mechanism in the cell to know how much you want to have this reaction occur. But again, it's like when we were talking about the temperature. Like some cell might function better with more of the product, some might function better with less. It all depends on the cell. So that's kind of where the Goldilocks hypothesis. You'll find like throughout this course, like when we go into pathways and we talk more about regulation, that it is all based on the cell's like way of functioning at its best and how it regulates the amount of things that it has in it. Does that sort of answer your question?
Yeah, thank you.
So yeah, this is just a visual to describe what I was talking about. So in general, enzymes, you want the high affinity for the substrate. Then the catalysis reaction occurs, and then at the end, the enzyme lets go of the product. So at this point, you don't want the enzyme stuck to the product. So then you have a higher dissociation constant, which is equivalent to a lower affinity, so that the enzyme is able to let go of the product. Cool. The next thing we looked at was enzymes. May, do you want to do this slide?
Sure. Okay. So enzymes, we have they're catalysts, so they carry out chemical reactions. You'll see that they will help carry out chemical reactions, make them quicker. That's kind of what a catalyst is. Enzymes are usually recognized, like if you see the ending ASC, ACE, like that's a pretty good indication that you have an enzyme. So enzymes are good for making and breaking covalent bonds. And that's kind of the chemical reactions that you're talking about. So peptide bond formation that links the amino acids together in your primary structure. The peptide bond is a covalent bond. Ribosomes also form so that's your ribosomes. Spliceosomes are going to break your covalent bonds between sugar molecules. So enzymes are also important to the cell because they change the chemical structure of the molecules. And this is, like I talked about earlier, this is your regulation. So you're going to either break the bonds or make the bonds depending on what the cell needs. One thing to note is that not all enzymes are made of pure like 100% protein. Some enzymes are made of an RNA protein mixture where the RNA is the catalytic component. And then we talked about this a little earlier with your ribozymes. So ribozymes are kind of a category of your enzymes, but you want to think of the ribosome as having RNA and protein, and that they are classified as ribozymes because they have that catalytic component. That's just your example for one that isn't 100% pure protein. And then that picture was just kind of like to reiterate what Michelle said earlier. You can see how enzymes kind of function, and then it also is another visual where you see affinity and the dissociation constant in place. I think we can go on to the next one. Does anyone have any questions for that?
More about enzymes. Enzymes have to bind to substrates for catalysis to occur. So they must have some affinity for the substrate, which you can see on the right. And then enzymes can undergo conformational shifts, which is kind of like shape shifting during a reaction, but ultimately they remain unchanged by the end of the reaction. And it's very important for enzymes to have high affinity for reactants in the products. This is just a repetition of what I said a few slides before. And it's because you want the enzyme to latch onto the substrate and then dissociate from the product. And these are just some more visuals for enzymes. Yeah. So does anyone have questions? No? Okay.
So why are enzymes useful? There are two main reasons: chemical reactions and cellular function. When you have a chemical reaction, the enzymes are going to speed up the reactions that take too long for the cell if they occur spontaneously. It might take too long for the cell to be able to function properly on its own. And because of this, since it speeds up reactions, it will increase the frequency of those chemical reactions. So for cellular function, it kind of goes hand in hand with the time of the reactions. So the time for the spontaneous reaction without an enzyme is usually too slow for the cell to function. So this is another thing of how it will improve the functioning of the cell. You don't want cells to be non-functional. And because of this, enzymes are able to be regulated. So cellular processes are also regulated. And when cell processes are regulated properly, the cell can function properly. So this will be something more present in when we get into pathways. Pathways are going to regulate cell processes, and enzymes are going to help them to do that. So it's a regulation mechanism. Like I was talking about earlier, it will be good to know like the enzymes can start and stop reactions when they need to. Does anyone have any questions on that? A big thing is function. Like, helps with cell function.
This is just a diagram of enzyme function. And it just shows the reaction progress over time and the free energy associated with the reaction. And yeah, you can see that without a catalyst, without an enzyme, the reaction takes a while to reach the transition state. And then with the enzyme, there's not as much height to reach a transition state. Yeah, so any questions?
So this is more of an explanation of the diagram you just saw. So like Michelle mentioned, there's a transition state. It was kind of that top peak. And all reactants must go through that before the product is formed. And something about the transition state is it's very unstable. Like the reactants don't want to be there. Like it's kind of like the next step before you become the products. And that's because it's a high energy state. So the activation energy that you need for the reaction to occur is the difference between the transition state energy and the energy of the reactions. And with enzymes, like I said, it will help speed up the reaction process. Enzymes are going to lower the activation energy. So it's lowering that higher energy state of the transition state. So it will stabilize it a bit more. And there's less energy for the reactants to reach that transition state. So stabilizing the transition state is what speeds up the reaction, and enzymes want the reactants to reach the transition state. So if you, Michelle, do you mind going back to?
Sure.
Yeah. So if you look at the solid line, that's without an enzyme. You can see that that curve is much higher, that peak. And then if you look at the dotted line, that's with an enzyme. Like it says catalyst on it, but the catalyst is your enzyme. That difference makes the reaction happen faster because less energy is needed for the reactants to reach the transition state when you have an enzyme. And then, perfect. Yeah, so then the free energy, that's your energy of the reactants. If that is greater than the products, the reaction is considered spontaneous. That's energetically favorable, wow. And then also exergonic because energy is given off in the reaction. If the free energy of the reactants is less than the product, the reaction isn't spontaneous. And it's endergonic because there's an input of energy that's required to create the products. So big thing here is definitely know that enzymes are lowering the activation energy of the reaction because they're stabilizing the transition state and lowering the amount of energy the reactants need to reach.
Yeah, I wanted to add something too that I should have said earlier in the diagram. So like when you look at this diagram, just imagine yourself if these are like two hills. So if you're on a bicycle, you'd probably want to bike the dashed line because it's a lot easier to get to the top of the hill. Can you hear me?
Cut out. Now I can hear you, but
sorry. What I was saying was to think of the two lines on the graph, think of them as you cycling up a hill. So you'd ideally want to go on the dashed hill because it's smaller because the transition state is stabilized there. So it's easier to bike up that hill than the one that's without the catalyst since it's a lot steeper. And so enzymes just make it easier for the reaction to run smoothly. And yeah, that's my analogy. That's what I wanted to add. And just remember, enzymes lower the activation energy. They don't change anything else, just lowering the activation energy. It's a big key idea and hint. And yeah, does anybody have any questions about that? No? Okay, moving on. So we have this question here. Why is it beneficial for the enzyme to lower the activation energy of the reaction? May, is this a question or is it like gonna say the answer next?
I put it in a question and then like if they have any answers. I can put a description in when we send out the slides.
Oh, okay.
I just like in the review to be more of like what they think the answer is.
Oh, okay. So why do you guys think it's beneficial for enzyme to lower the activation energy of a reaction?
There are multiple things. So if we can like start a discussion or use the chat. All right. So I see a lot of the reaction could go faster. That's good. That's one of them that I can think of on the top of my head. Does anyone else have anything? Just say like yes or no, or someone put it in the chat, so it's not like.
I can't quite see the chat because I'm screen sharing, but sounds like good things.
All right. Yes. Okay. That was pretty good. So energy efficient. Yeah. So you guys like got the two main things. So yes, the reaction is going to happen faster. If you need products, that is great if you have an enzyme because you have less time that you're reaching the transition state, the reaction can happen faster. Also, it is energy efficient. So when you think about a cell and it's trying to carry out all these cell processes, and you want them to happen, and you're trying to regulate everything, the cell doesn't want to use a lot of energy to have that happen. It wants to be as efficient as possible, get everything done with the least amount of energy, because in the cell, if you have a functioning cell, energy is costly if you use too much of it when you don't need to. So that's why it's also beneficial, because if you have an enzyme, then you're not going to, it's like if you have an enzyme and you're doing that same reaction, like that could be done without an enzyme, then you're taking away like the time it takes, you're taking away the energy that you need to get to that higher energy state in the transition state. Does that make sense? Yeah. Okay, cool. Yeah, so energy efficient. The cell wants to work as efficiently as possible. It doesn't want to waste energy because energy is really valuable. Then, like what we talked about a lot already, it's just going to be a faster reaction because it's going to take a shorter amount of time and a lesser amount of energy to reach that transition state. So good, awesome. I can put that in feedback, but right. Michelle, do you want to do that one?
Oh, okay. So then we talked about how enzymes work. So there are three main ways they can work. Enzymes can bind to the substrate directly and arrange it so that there is a way to encourage the reaction between them. Then the second way is binding to a substrate, and it rearranges the electrons within that substrate, creating positive charges and partial negative charges that favor reaction. Then the third way is that the enzyme can strain the bound substrate, forcing it towards a transition state to favor reaction.
Something I want to mention on this, like I know we talked a little about it in class, and some of them aren't very clear on what happens. Just a hint, this has been an exam question. It's like commonly an exam question. So know that these things happen. That's a big thing. I don't know, Michelle, was it on your exam?
Yeah, I would say there was like a question about it.
It was like exactly like what can an enzyme do to lower like activation energy?
Yeah, just have the ideas in mind, because there was one that was like what kind of enzyme do, and it was similar to that. Also, remember, I'm gonna say it again, enzymes only change or only lower the activation energy. They don't increase anything else or decrease anything else.
We can add that to a slide.
Yeah.
Because that's a big idea. So yeah, know these figures, know what they say. That's your hint.
Yeah, I can almost guarantee there will be a question probably about, does an enzyme lower activation energy? Does it increase Gibbs free energy? Like, what does it do? And then you'd say lowers activation energy. And there definitely was that one. Okay. Next, we're going to talk about regulation of proteins.
Wait, there's a question in the chat.
Oh, sorry, I can't see the chat.
Yeah, it just says, what type of question could be asked about figures like that? It's like, what is it? It would be very straightforward. It would be.
I think it would be a straightforward question.
Let me see. If I can find
Whoever asked the question, I can look more into it at the end of the review and let you know like how it's asked, but it's usually not asked in too much of a difficult way. It's in a way you'd know. At least for me, when I did it, I think it was like a multiple choice, like or like either like a true false or something.
Does anyone else have other questions?
No questions? Okay. Okay, I don't see any other questions. I can see the chat. Okay, so next we're going to talk about regulation of protein. May, you want to take away this slide?
Yeah, so we briefly talked about allostery. The main idea with this is that it's the binding of a molecule to one site of a protein other than the active site. So when this happens, if something binds to an enzyme that is not at the active site, that can sometimes induce a conformational shift. So if something like, say you had a like sponge or like a Pac-Man shaped sponge. If you put the sponge like on the other side of Pac-Man's mouth, then like that could change the shape of this other sides of Pac-Man, and then that might affect the part where something is going to bind into the Pac-Man like shaped mouth part. So basically, it can induce the conformational shift where now the site where the enzyme usually binds a molecule, it might not fit properly. And that is going to affect the enzyme's ability to function and perform its job because it isn't able to bind as efficiently. So Laura is big on structure-function relationships. This is one big main idea thing, an example of it. So if something affects the structure of something, how does that affect its function? If there's a conformational shift, there's no guarantee that the enzyme is going to be able to bind the molecule it's supposed to and perform its job there. There are different binding sites and types of allostery, but we didn't really go into it. No, basically that there can be conformational shifts because of another molecule that binds somewhere than the active site. And then that conformational shift doesn't guarantee that the enzyme is going to function properly. Does that make sense, or does anyone have any questions for that? No?
Just one.
Oh, I'm sorry. Was there a question?
Yeah, just one. Before you said there can be conformational shifts. Did you say that that guarantees that the enzyme won't work properly?
No, it does. It like
It may.
guarantee.
It might.
Yeah.
Okay.
Depending on the situation. It's like when you guys do questions about if I switch this amino acid for this one, could it potentially affect the function of a protein? Sure, it might if you picked an opposite amino acid, but if you picked one that was similar size in chemistry, then there's a chance that the protein could be fine. It's just kind of like that. Based on your reasoning, you could infer that depending on chemistry and size that it would be fine. It's sort of the same idea here.
Okay, I understand. Thank you.
Depending on like situation to situation in a question. Yeah.
Think of like, you know how if you press down on a balloon and it doesn't pop, but it like changes shape?
Yeah.
Yeah, so that's kind of like if something were to bind to an enzyme other than the active site, like there's that shift, there's the possibility of a shift in the enzyme shape. So in the enzyme shape where it normally would bind another molecule, because there's that shift, you don't know if that shape is still the same as it originally was.
Okay, I see.
Like since it's not like if there's a conformational shift, since the shape changes, you don't know if it's going to be able to bind the same molecule that originally was supposed to.
Got it.
Cool. So any more questions?
You can try to find another picture and put it in later.
Okay, I can't quite see the chat, so I'm gonna assume there's no questions.
Yeah, I think the chat's good. There's no.
Okay, so next we talked about post-translational modifications. And I did receive a few questions about these types of modifications, but it's important to know that they occur after the protein has been made, hence the word post. And this usually occurs after tertiary structure, because remember, when you think back to when we talked about tertiary structure, that is your overall shape of the protein. So that's when your alpha helices and beta sheets from secondary structure, they all form together to make this massive protein. And for post-translational modifications, these can often be covalent modifications and functional groups that are added to the protein. And this process is regulated by enzymes, and enzymes themselves are regulated also. And these functional groups can be tacked on to the end of a protein, and then they can also be removed. And essentially, the post-translational modifications is like a switch, because when you tack on a functional group, or when you make a post-translational change, you're changing the protein surface chemistry, and you're changing the structure of a protein. And when you change the shape of a protein, you're changing its function. And by removing the group, you're also doing the same thing. You're changing the chemistry of the protein again, but then you could also affect its function. And so on the right here, we have, this is P53. It's a transcriptional factor. And this transcriptional factor is heavily post-translationally modified. And Laura talks a lot about how surface chemistry of something speaks to its function. So because there's different groups on there that are tacked on, you can see in yellow, there's some phosphorylation functional groups, there's some ubiquitin groups in green, and then in blue, there's acetylation, and they're all kind of scattered into sections. So you can see that the chemistry of it kind of alludes to the function of it. And yeah, so based on whatever group you have, there can be different functions. So you can see that maybe one group can move things to the proteasome for degradation or move to the nucleus, like there are different functions of P53. So yeah, that's post-translational modifications. Does anyone have questions? No? Okay. And next we talked about nucleotide binding. And this leads to different binding and active states. And this is when we talked about GTPases and ATPases. And these are enzymes, which are also proteins, that can bind to GTP or ATP. And when you tack on a nucleotide to a protein, it gives you the ability to change or cleave bonds between phosphates and change GTP to GDP. And usually results in the conformational change, so the shape shifting, and that leads to a change in its function. And this GTP-GDP switches for the protein are regulatory mechanisms. And much later on, we're going to be looking at signaling pathways. So there'll be JAK-STAT pathways and survival signaling pathways. So this will make a lot more sense. Also, when we look at nuclear import and export, you'll see how GTP plays a role in activating proteins, GTP. And then when the protein is bound to GDP, the protein kind of shuts off. So yeah, so I tend to think of GTP and GDP as light switches. When they're bound to protein, it either activates or deactivates them. So GTP, you can see in the image below, the GTP is tacked onto the green thing, which I think is supposed to be protein. Yep, it's supposed to be protein. It turns it on. And then GTP is, I think it stands for guanine triphosphate. So then when the phosphate is cleaved, it becomes guanine diphosphate, so with 2 phosphates, and then the protein is off or in an inactive state. So yeah, that's it for nucleotide binding. Does anybody have any questions? I can't see the chat, so are there no questions in the chat?
Yeah, there's one.
as well. Can someone read it to me, please? I just don't have access to the chat because I'm sharing the screen.
Oh, and the person sent it to me privately. It says, what kind of bonds do GDPs make with proteins?
What kind of bonds? I'm not too sure if she dives into specifically the bond that GTP makes to a protein.
Oh, I get why they asked this, because there's a question on the practice set that actually asked something like this. It was asking what type of bond does aspartic acid make with GDP? So does GDP have like a charge or something, or is it like uncharged? Because I don't think there's any way we can answer it without knowing that.
That's a good question. So let me see. So I think for GDP, I think it has its own chemistry. So I think what she'd want you to do for that is look up the function, the what it looks like chemically. So can you just repeat the question, please?
Yeah, it's what kind of bonds do GDPs make with proteins?
Yeah, so I think for that question, because it has its own chemistry.
It's the one with the figure, right? It gives you GDP in a binding pocket. Is that what it is? Yeah? Okay. So that you can sort of think that's kind of like the cyclic AMP figure that we looked at. That one, it's a question about bonds. So if you look at the structure of GDP and where aspartic acid is on that, you want to look at where it is, and you want to focus on the orientation and what type of bond it will make based on the chemistry. Like the chemistry is given to you in the image. So if you look at what's projecting off of the GDP and what's projecting off of the aspartic acid, where, like, based on the orientation that you're given with the binding pocket and the GDP, what bonds are going to be made between those two things.
Yeah, so for a question like that, she doesn't expect you to memorize what GTP looks like or what bonds it makes. She's just trying to give you an example of something, and then she would provide an image for you if it was an exam. And then based on what you know about the molecular biology or the chemistry of the molecules, you can figure out what bonds does it make. Does that make sense?
Yeah.
Cool. And I actually think this is the last topic for the review, so that is everything.
Do you mind if I share? I found an image that might make Alistair, like whatever I was saying, a little easier to visualize. Can I just share that one thing?
Sure.
Okay. That might make it easier. So this is kind of what I was saying. Can you guys see that? Not this. Can you see this picture right here?
Yeah.
Okay, so this is kind of what I was talking about with that whole balloon thing. So this is your enzyme, and here's your active site. This is how the active site is normally shaped. Then you have this other site that's not the active site. That's going to be like your allostery. That's where something is going to bind other than the active site. And if you look at the bottom of the arrow, when that binds, that active site shifts and changes shape. So by the time when the time comes when the substrate needs to bind the active site, because that has closed up and the enzyme, like, it doesn't necessarily have to close up, it could have been made bigger. Just because it changed, that substrate can't fit the way it properly does. That's what the one mechanism she was talking about kind of means. It's because you have something else binded to the enzyme changes shape, and that doesn't allow the substrate. There's the potential for the substrate to not be able to bind to that anymore because of the conformational shift. Does that make a little more sense?
Yes, it does. Thank you.
I can put this image in the slides like later before we send them out, but Hopefully that makes it a little more clear.